Hello, everyone. Please take your seats, and we're gonna start with our next panel, which is exploring ARPA-H longevity projects. Very exciting. And we have three great program managers, ARPA-H program managers with us today. We have Andrew Brack, Gloria Elliott, and John Hebert. And, before we get into talking about their programs, I just wanted to address the question of how is ARPA-H different from federal government grant programs? Because I think a lot of people get confused about this. Maybe it was covered yesterday, but not everyone was here yesterday on the panel where ARPA, I think Andrew was on the panel yesterday. Maybe you covered it. Maybe let's just briefly cover it, because I am guilty as a Silicon Valley. I'm Sonia, and I am chair of the Alliance for Longevity Initiatives, and I'm also an investor. I invest in longevity biotech companies, and I see some of my companies here today. And even I get confused. I'm like, oh, it's an ARPA-H grant, but it's not a grant, is it? So does one of you want to explain how it's different?
Yeah. Okay. So they're not grants. So NIH plays a fundamental role in discovery, exploration, and setting the groundwork for discoveries that can one day be translated to people. But ARPA-H is there between that and where VCs might pick it up, where it seems commercializable. So it's still a sort of early idea or concept that we can see might have a huge impact on healthcare, but then is it, it's going to have a lot of technical risk, so no one else is going to want to pay for that. Mm-hmm. That's one aspect of ARPA-H that's different. There are other aspects as well in how we operate that are very different. We're there as for only a few years, term positions, because we're given a lot of autonomy. And maybe you guys want to add to that, but that autonomy also makes us very different.
Yeah, it's sort of I think I've got the best job in the world. And the reason for that is you get this idea, anyone who's formed a company, you've got this idea, and the big problem is you need to raise money. Well, as a program manager, we have the idea and the money. And so then we're going to recruit people who can execute on the vision that we put in place. So that's why it's not a grant, right? It's a contract that says, here's how you can change healthcare, change the world for all Americans, but you've got to do these tasks. Are you able to execute? Are you able to take those risks? And every quarter, there are milestones that need to be met. They're aggressive milestones. If you meet the milestones, you get paid. If you don't meet the milestones because either you want to go at your own pace, you do not get paid. Sometimes science pivots. We don't know the results of an experiment until it's done. As a program manager, we work with the teams to pivot to find the right answers to that science. So it's very active in the process as well.
I like that. That feels very efficient. Sorry, do you want to say?
No, yeah.
Gloria?
No, I was just gonna, double down on that it is an active, managed project. So we're at the table with you. We're invested in your success. We're looking for opportunities to get you to the end game. So we wrap around you, and if we need consultants to help guide next decisions, as Andrew said, sometimes we need to pivot. Sometimes things come up. It might be policy. It might be some regulatory thing that has shifted. It may be that there's been innovations in other space that have happened in, the first year of the project, and we take in all of that, and we are at the table with you trying to drive to a good end game for the American public.
I love this. I love what you guys do. So why don't we jump into, maybe if each of you can explain the program that you're working on. You want to start, Andrew?
Sure. So we built a program called Prosper, kicked off about 4 months ago. It was $144 million that went across 7 teams. And it really addresses the problem that we're all aware of in the room, which is we all want to develop therapeutics for aging, for health span, yet there's no regulatory path. And the ability to do that would take 20 years to run a trial, billions of dollars. It's not financially possible or realistic. And there's also no regulatory path to being reimbursed, as Dr. Oz mentioned and Lowell mentioned just previously. So the program is designed to build that regulatory path, the train tracks, so that we can, after Prosper is finished, everyone in this room can build a therapeutic for health span. So how do we do that? Well, it's really about shortening those clinical trials. As mentioned earlier, surrogate endpoints. So we're going to generate clinical endpoints that are feel, function, or survive that allow one to test their therapeutics in a sort of building the data first paradigm. We do that with three different interventions. One, the FDA will require sort of some level of intervenability. And so that's going to be through gold standard exercise. 18 months exercise trial. Can we move a clinical endpoint, which we're framing around the concept of intrinsic capacity? This is a WHO-defined composite measurement that captures feel, function, and survive. And we know it does those things because people who have low intrinsic capacity die sooner. They get diseases earlier. So this is one way of getting that surrogate, but we have to build the data, and that's what ARPA is unique for. The second part of that is, how do you bring a therapeutic to market and test in people who are not sick? Well, you can use drug repurposing for that. So GLP-1s, SGLT2 inhibitors, rapamycin, they're going to be tested. 3-year trial in aging, non-diseased people. Does it change intrinsic capacity? And finally, if the hypothesis is right that we can shorten duration of clinical trials, we can test new therapeutics. So we brought 4 new teams with their own biotech drugs that have never been tested in humans in aging before and ask, can we now move intrinsic capacity in an 18-month trial? If successful, we've proven the hypothesis, we've got the data for the FDA, and the train tracks have been built. That's Prospect.
All right, John?
Yeah.
Do we want to? Yeah, let's go, John, and then
Yeah.
Gloria.
Yeah, so Andrews is a great example of what makes ARPA-H ARPA-H. This is not something that could be done by NIH or any others. And so our program, I think, is similar in that sense, and our portfolio, my team's portfolio at ARPA-H. And all of us, I think, relevant to the audience here today, are taking approaches to aging and longevity that are not necessarily easy or possible to take otherwise. We heard a lot earlier about prevention, education, access. Those are all things ARPA-H is interested in health in general, and that includes aging population. But also, we have as a priority at ARPA-H to directly address aging itself. And so that's where we come in. And the program, which is part of a broader portfolio that I'm excited to share with you now, is one where we can address any type of brain damage. So that could be from stroke, Alzheimer's, trauma, or aging itself, which, sadly, all of us in this room, our brains are slowly shrinking. So there's damage, even if we're very healthy, that I think at some point we'll want to address. And so we're doing this by following what nature taught us about how the brain develops. We know that it gives rise to a product that can do all the things that we want it to do, store our memories, allow us high levels of cognitive function. So we're using that as a blueprint to regrow new brain tissue in our adult brains. It sounds crazy at first, but when you dig down into the technicalities of what you need to overcome to achieve that, I think we have a shot at it. And this is also why it exemplifies an approach at ARPA-H, where at first these things may sound crazy, but then when we dig deep and do the diligence, which we did for more than a year on this, like, how could we actually make this work? Then we come up with a plan, and we execute, and we partner with people like you out there to make it happen. And, yeah.
Right. Gloria?
Well, I round out the panel in that Andrew is very much focused on the patient and early detection and early treatments, and John is working on these novel tissues and replacement tissues when there is a need for an intervention, if from stroke or some kind of brain damage. I'm very much focused on ensuring that everybody can access those advanced therapies when we've discovered them and develop them to the point of of integration. So I'm in the Scalable Solutions office, and our mission is very much to drive access and to reach more people. So my programs are, I have two that are on the street now, Boss and Tiger, and they are complementing programs that are targeting access. So with the Boss program, we're looking at the cost and logistical complexity of cell and gene therapies. So right now, cell and gene therapies need both the starting material when you send the blood product to the manufacturer to create the product that has to be cryopreserved, which is very logistically complex and adds a lot of cost and adds risk to the starting material. And same thing on the other side. Once the cell therapy has been manufactured, they've got to do that process all over again, cryopreserve those cells, put them in a liquid nitrogen shipper and send them to the hospital. That limits the type of medical facility that can actually give that therapy. So generally restricted to well-resourced big hospitals. So the BOSS program is taking this audacious 200-degree leap in temperature. We're not just trying to increment, we're really just trying to turn, this is what's called cryochain or cold chain, turn cold chain on its head and get all the way to ambient storage. So we just wipe out all this need for refrigeration. The Tiger program, it's an exploratory topic designed, it's targeting more tissues, like the front program is working on, and organs. We're all, I think, very familiar with the problems we have with the organ shortage and the short shelf life out of the body and the need to rush everything to get to the end user. So the Tiger program is focused on new innovations to try and address that time and temperature issue. Can we store organs for longer? Can we develop the technologies that allow us to biobank and stockpile organs? Can we improve? Can we, just like BOSS, can we find practical storage temperatures for organs that don't require liquid nitrogen? So all about access and bringing costs down and making sure that all Americans can have access to these advanced regenerative therapies.
Yeah, you were all very complimentary, actually. You sort of all go together. Was that by design, do you think? Or does ARPA-H just sort of go through different, sorry, I'm jumping around in the questions, but I think this is a perfect time to ask that question.
Yeah, I can take that. So, we are consulting on a daily basis and putting, metrics into each other's programs and tightly coordinating these programs. But I would say we're all on board with the mission, and we're aware of the problems, and we do talk about that, throughout the agency all the time. We know who's who at the agency. We know, what our individual passions are. We know what piece of the problem we're owning, and then we're all driving towards that same outcome. So I think that focus on the end game, inadvertently coordinates the
I think there's also the program manager sort of mentality is we don't know the answer. So why are you going to bet on one solution? Everyone can have an opinion. We've got to do the work and find out the answer. And so I think from an ARPA level sort of perspective, it's take multiple shots on goal. Mm-hmm. And are there along those ambitious sort of programs, short-term wins along the way? So we may not get to the sort of North Star, but we'll find an intermediate could also have a major impact. And so that's, I think, why by nature it's complementary.
And we're constantly pivoting and talking to each other and learning and converging when it makes sense to converge and pivoting when that's the better solution.
Yeah, so to some extent by design, we're complementary to fill the longevity needs or in regenerative medicine in this case and drug discovery and application. But it's also a somewhat operationally forced by ARPA-H because we're a small group, we're a small agency, we're super lean. We have a lot of responsibility to the public given the size of our budget, but we're very lean. And so by design also, we don't overlap in what we do. It wouldn't make sense. And so that's part of what we do.
Yeah. Right. Yeah, that makes sense. Okay, let's go back into some take a deeper dive into the programs then. Back to Prosper, Andrew. I'm really interested in this idea of intrinsic capacity. Can you say more about that? And, a lot of your trials are happening in living rooms rather than in the clinic.
Mm-hmm.
And, how do you validate biomarkers, and what does the trial really look like? And can you say something about that?
Sure. So we made a bet on intrinsic capacity being the thing that we could put in a clinic. And so, but we also know, we think healthcare is going to move at home, especially if you're not diseased. You don't need to go to a clinic if you're just 55 years of age versus 40 years of age, right? And so we thought, why don't we try and marry the sort of clinical endpoint to an in-home kit? And so intrinsic capacity is this composite measure. It takes feel and function. It has locomotor, vitality, cognitive function components, the things that decline with age, right? And so let's say we generate a score. And you can do the first version of that with a sort of large data set like UK Biobank. The challenge for the performers who are building the intrinsic capacity is we've asked them to harmonize basically all longitudinal data sets that are out there. And so that's the first challenge. So we'll generate this intrinsic capacity score of probably over 1 million different populations. That's version 1. And so that's going to be run in clinical trials or in a lifestyle sort of trial. Because we need to get that to the FDA. So here's the data set. We know it declines with age, but is it intervenable? But at the same time, wouldn't it be great if everyone at home could have a validated kit? We spoke a lot about biomarkers yesterday. We spoke about things in home. It's no good unless it's validated, right? You need data, you need intervenability. More importantly, the FDA do. Mm-hmm. Otherwise, we get gray market stuff and people pay money for. It may not do any harm, but actually does it provide value? Mm-hmm. And so we were determined to build something that could be regulated by the FDA and could be in-home, so people say when that measurement moves by a certain amount, it's actually clinically meaningful because A, it changes with exercise, B, it changes with rapamycin and GLP-1s. Mm-hmm. So what does that take? Large trials, a lot of money, and years. So it's not easy, but that's what the privilege of this position allowed us to build.
How far do you feel like you are away from it?
Well, so the program is started 4 months ago. But we have put our first COA in, a clinical outcome assessment to the FDA. That's now under review. We put a white paper out, consensus review on why intrinsic capacity has a shot. We don't know the answer. We've got to build the experiment, do the experiment. I had a conversation with CEDA, CEBA, CDHR, all about this in the last couple of days. So it's moving forward. But until the trial start, we don't know. The first version, based on the UK Biobank, that data should come back in about 8 months' time. So that'll be the first time we could, in theory, get a kit in people's hands.
Wow. That's actually not bad. That's a decent timeline.
12 months.
Yeah. That's great. Okay, let's jump over to Gloria. Can you tell us, what is the thing from nature that nature might have already solved that gives you confidence that you could have this 200-degree leap that you were talking about? How is it possible that it might be achievable?
Yeah, nature's already given us a prototype. There are so many species in nature, they're called extremophiles, and there's a specific class of extremophiles called anhydrobiotes that are actually able to dehydrate and enter into a glassy state and stay in suspended animation in that state for years. And when conditions are hospitable, they can rehydrate, they return to full function. So these are species that some of you may be familiar with, the water bear, the tardigrade. You see lots of pictures of brine shrimp, Artemia. There's a water midge that's really special called Vonderplunky. So this is a species actually that Japanese researchers have isolated some cells from, and they've been able to grow a cell line culture that is also that retains the same phenotype. It's still desiccation tolerant, and that's really enabled them to learn about all of the organization that happens in the cell in response to dehydration. So there's no new physics needed, no new biology. We've seen it happen. It's out there in nature. We just have to understand it well enough. And I think this cell line has really enabled us to have a really a good look into intracellular reorganization. So now the challenge is bringing together the right experts. So we need material scientists, we need biologists who understand cellular reorganization, we need process engineers to build the process flow and the system that would enable us to control the dehydration conditions, or maybe it's some other approach. It doesn't have to be exactly like nature. Really what we want to do is get to the same end state where we've got this really confined, suspended animation state, so we can hold things at ambient. And we put a lot of constraints on the program because we don't want to create something that's more costly than cryopreservation, which is easy to do, right? When we get inventing and developing, it's easy to just chase the science and not keep your eye on the cost ball. So we built that in. Those metrics are in the program. We need, if you're going to build a system, the system should be affordable. The product should be easy to use by the end user, and all of those things. But yeah, I think we know how to do it. It's a matter of whether we can converge it all and get to the end. I think we can.
Proof of concept. And then Jean, I feel like you have the hardest program because you're regrowing parts of the brain, and I mean, and then you kind of hinted at like you could potentially like regrow anything, right? And so what in running your program, what kind of things keep you up at night? Like what are the hardest things you're dealing with? Because I feel it's really hard.
Yeah, sure. Technically, there are a lot of things that keep me up at night that I think about, sometimes for weeks, but have always, with the team that I work with, and now the external team that we're contracted with, we can troubleshoot these things and overcome them. So there isn't a problem yet that we haven't thought of a solution. We don't know if it'll work. Again, as Andrew says, we have to do the experiment to see. But the brain is complicated, and there's a reason that currently there is no treatment for what we call permanent brain damage, which is hugely costly to the society here in the U.S. and worldwide. So there's a tremendous need there, and I think it's worth the challenge to think about these. The specific technical hurdles that we think about is to really do what we need to mimic normal development. Again, that's the only blueprint we have for success, how the brain normally develops. And we have a relatively deep understanding of what needs to happen. And so at the same time, that sort of points us to the things that could go wrong in our case that we have to focus on. But yeah.
Well, this leads to the next question then actually for all of you is, you're only there for, 3 years, and then maybe another 3 years after that?
If we're invited back.
And you're doing really difficult things. So they're obviously not all going to work out magically. What does success look like if, like, in each of your programs, what does it look like? Maybe you don't get the complete end goal, but what are the goalposts in between that you can still say, hey, that was successful?
So in my case, I think moving the needle on the cold chain. So getting people out of the mindset that this is the only way we can sort things. So I think when we had the COVID-19 crisis and we rolled out those first, I think it was the Moderna vaccines that required -80 storage. And we were just obsessed about installing as much infrastructure as we could for cryochain, cold chain, so that we could distribute all of these vaccines. We were in crisis mode. But I think that just got everyone on this wheel of we just need more refrigeration. So I think if I can disrupt this field, if we can have enough success to show that we don't have to do it this way, there are other ways to store things that doesn't require such a costly infrastructure. I think any breakthrough, one cell type, at ambient storage for some period of time, I think that's really going to open the eyes of the industry, and people are going to want to get on board because they see it's possible. And if it's possible, they don't want to be left out of that game. You don't want to be stuck refrigerating your stuff if there's an alternative that you can leap out of that mode.
Right.
Yeah. So I guess the biggest thing would be for people to think about brain damage as no longer permanent. Right? We take that word out of the sentence permanent brain death. So a proof of concept showing that would be a great outcome of this program.
Where is that most likely, you think?
What's that?
Where is that most likely to happen? A proof of concept and
So we have very high standards for showing that the new brain tissue would function normally, and so we would have to meet those standards. So we have preclinical tests where we could silence the new tissue transiently to show that whatever function was reacquired gets transiently lost, for example. So technical milestones that way that would demonstrate proof of concept, and that's a game changer. That, I think, changes the world in a lot of ways. So that ideally is the case. We also, to get there, I think we'll need, and this is part of the success mentality, is to fail often and reiterate often. And so that's part of our plans as well, and we'd like to be able to do that very quickly and efficiently to get us to our goal. And then, also just in doing that, I think we create a broader industry that now realizes there's a lot of different parts of the brain that get damaged for a lot of different reasons: trauma, strokes, degeneration, tumors. There's a list there, and right now there's nothing we can do about it. And so breaking that open, having a new industry address that, including for aging. Again, all our brains are shrinking, and function is diminishing. And so sadly.
I hate to be
These are
reminded of that.
Yeah, but, so I think there's definitely going to be a growing need as we get healthier and live longer to address this problem. So we need an industry that can back it up. Ultimately, if we can just spur that on, that would be great.
Andrew, what's success for you?
So the big success is we build the regulatory path so that everyone can test drugs for health span. That's the thing. Do we get that in 5 years? That's going to depend on the results and the FDA aligning with that. That may take 7 to 10. Right? The partial win is that we will integrate intrinsic capacity, age-associated decline in intrinsic capacity, as a measurement in various trials over the next few years. I encourage you, anyone who's writing ILBs right now, please put them in your trial. The second part of it is we will know whether rapamycin, SGLT2s, all the repurposed drugs, plus the other new drugs we're building by the teams actually improve health span. We'll know that. I think that's the short-term win.
Yeah, that's great. Okay, let's transition into a question. Are we still ending at 11:30?
11:32.
11:30. Okay, so I'm just trying to keep us on time here. So each of you came from academia or industry, and you can really see that, by the way, right? You're not longtime regulators or something like that. And so now you're placing bets on other people's laps. So you're on the other side of the table. How did being on the funding side change how you think about the research you used to do yourself? Would your past self have gotten an ARPA-H contract, do you think? And what kind of advice do you have for people?
I think what's important is what Gloria said earlier. This is a partnership. We work closely. I talk to my outside people, like, if not daily, like multiple times a week. Okay. So I am doing essentially what I was doing when I led an academic lab or had my own company. We're in there with the performers doing the work. So it's not that different, except now we're also the funders, the VCs,
I think if I were to go back to my academic life and look at what I know now versus what I knew then, I think I really underestimated the role of a team integrator. So as an academic, you do teamwork, but you also, I think, feel the pressure to be an independent investigator. They use that word a lot. So writing your own grants and doing your own thing and establishing your own little world where you're the expert. But really, I think the ARPA level problems get solved by the community of people that come together to work on it. And so the role of the integrator, the person who can look across the whole complexity of that problem and say, gosh, we need an AI guy, we need a person that knows regulatory, we need someone who's got the patient perspective, who can give us the profile of what we need to build, and on and on and on, that can look at the complexity of the problem, not be intimidated by it, go get the team, get them all working together and lining up and going in all in the right direction and speaking the same language. I think those people are special and rare, and I think it's what it takes. Like you really need that coordination, that maestro, I call it. You need a maestro in the lead. So I think I didn't value that as much as I do now, sitting here in this role.
Yeah, I think the role as is basically a technical founder with a team of 20 or 30 people. And so I think that's the mindset, I think, mostly. Would I have gotten it? I would not have got an ARPA project proposal grant when I was writing grants at MH. No. But I probably could have got one when I found my company. Because I think the conversion maybe to the recommendation out there, if anyone's trying to submit proposals, think of it as you're building out your company. You've got to have milestones, deliverables. And like every company, you want to change the world. That's the sort of thing we will fund. What we don't tend to fund are wishy-washy things that we think is incremental or could be done by others.
Mm-hmm. Mm-hmm.
Yep. Oh, and also, we are recruiting PMs. So there's 35 of us in the agency, large budget. So if you want to come and join us, change the world with that sort of technical founder sort of mindset, come and chat to us.
Okay, perfect. Yes, I think there probably are some people here who want to do that. Let's go back to the regulatory framework. So you've put this together. How likely, what does the path look like? I mean, how do you see that coming together?
Okay, how do you see it come together? So there's a couple of different things, at least with Prosper. There's the device lane. There's building the data for the endpoint, intrinsic capacity. So that's just requires data, and a change in that score is clinically meaningful. So i.e., if my score is 20% lower, I'm more likely to die or get a disease. So think about it like pediatric growth curves, right? Well, this is like an adult health curve, where at some point you'll reach a threshold and bad stuff's going to happen. So we have to build the data so the FDA say that number matters. And it also has to be affordable enough for the test, so CNS matters. A big expensive test, a DEXA scan, is not going to work. And so they're the things we think about for regulatory approval and deployment. In terms of the drug space, there's David here. There's two possible ways to go. One is we need to change the framework. So it could look like a sort of multi-morbidity endpoint. That's kind of tricky sometimes because you can end up just making a cancer drug and it just actually moves and get the wrong label for the indication. FDA don't like that. Accelerated approval is a thing for frailty, for example. Unmet need, cost, and then you back end that like obesity drugs have done with BMI. So frailty, I think, is a good opportunity for accelerated approval. And frailty looks like aging in every other way, except it's more exaggerated. But you've got to do the experiments. So with Prosper, will we end up getting to that approval point? One could imagine we get engagement, but the FDA say, do another experiment. A 10,000 person type of experiment, and that's not what ARPA will do. We leave that then to the larger companies.
And do you talk to them very often? Like, are they excited about what you're doing, or?
FDA?
Yeah.
Yes. I mean, I talk to CEDAR and CDHR about the devices. I talk to CEDAR about the drugs. They're thinking about this actively and positively.
Nice. Okay, good. I'm happy to hear that. Sorry.
I think the advantage, so I think your job is a little harder with the FDA than with regenerative medicine approaches. And, they're pretty convinced those have high potential impact, and they all have great initial disease indications to address. And so in that sense, we have less to prove to the FDA. They're pretty much on board in that sense, at least. we still have the costs and everything else to address, but yeah.
I don't think
I think that covered it.
Okay, so in the next 5 to 10 years, if at least a lot of your, can this be the last question? If a lot of, if you have some successes, what does this look like for the average 80-year-old American?
Yeah.
Start, and then we'll go.
I mean,
Yeah.
I'm losing my dad to cognitive decline. I mean, he has dementia, and you know, I'm saddened by like we're going to be 5 to 10 years too late. Like this is something that I think we'll be able to address in that time frame. So that's true for lots of people. I'm sure you know others have people like that. So yeah, I mean, it's going to have a huge impact on people's lives.
And I would say that to bridge from that, you know, if we have those therapies, we want to make sure that people can get them in their hometown, where they live, where their family is. If you're at end of life, you don't want to travel, you know, across the state or across the country to a big medical center and spend, you know, your last chance at life sitting in a hospital getting an advanced therapy that can only be delivered in a hospital with a medical school. So I think ensuring that people can get the medicines they need at home, where they live, you know, where their loved ones are. That's how I see my programs impacted.
Yeah, and again, PROSPER is about prevention. And so I think we spent a lot of time in the last couple of days talking about disease. There's a window between being optimal fitness and disease. It's about 40 years. I want PROSPER and the advances we have to give optionality for those people in that 40-year window. Like, can we maintain that optimal health before we even consider getting a disease?